Um, so as Dr. Waldman mentioned, I am the Director of Research Safety and Biosafety here at OSU. I work over in the Environmental Health and Safety Office. Our offices are over on West Campus, over on Kinnear Road. So if you ever need anything, feel free to contact me or anyone in my office. My contact information is at the end of the presentation, so if you ever need anything, you know where to find me. There are a couple training programs that you need to take online. I think they're mentioned in your syllabus, but I'll let you know what they are. You need to take the bloodborne pathogen training. There are two, if you haven't already done it, when you go in to take the training, you'll find two training programs. One is the initial training, one is the refresher training. If you are brand new to the university, which I imagine most of you are, you're going to take the initial training. If you have been here previously and worked in a lab and you've taken that initial training before, then you can take the refresher training. So it just kind of depends on if you've ever taken the initial training. The second one is the lab standard training. The lab standard training covers all of the chemical safety that you need to be aware of when you're working in the lab. I am not going to cover that stuff today. I don't want to be redundant. I don't want to tell you all the stuff that you're already going to learn online. It's boring enough as it is, so I don't want to go over it all again. But if you go through that and you have any questions about anything in the online programs, please don't hesitate to call me. I'll be happy to answer any questions. When you do the training, you're just going to go to our EHS website, and if you click on safety training programs, it will take you to a web page where you can get to the login page. You just log in with your OSU name.number and password, and then you can get to any of the safety training programs from there. Okay, so today and tomorrow, we're going to spend some time talking about these topics. Today we're going to focus mainly on the first two or three. Depends how much time we have. We'll be out of here today by 9:30 because I have a meeting that I need to be at by 10 o'clock on the other side of campus. And then we'll finish up tomorrow. We might go a little longer tomorrow, but I'll have you out by 10 tomorrow. So today we'll talk about biohazards and biosafety, biosafety levels and animal biosafety levels, also risk groups, what are the different risk groups of agents. Then we'll talk about engineering controls, work practices, personal protective equipment, hazardous waste disposal and sharps management, and then a little bit about occupational health, medical surveillance, what you do in the event of an accident or an exposure, and a few little additional safety issues that you might need to know about. So let's start out with biohazards and biosafety. You guys are going to be working in, obviously, a biomedical environment, biomedical laboratory. You will obviously be working with some chemicals, which you will be learning about in your lab standard training. This module that we're talking about today, we're going to focus mostly on biosafety. Okay? And biosafety is all about working with biological agents. The big hazard here, obviously, is biohazards. What are they? We need to know what they are to know what the risk is. And biohazards are basically any agents that are derived from biological agents that are capable of producing disease in humans, animals, or plants. The ones that we are mostly concerned with are the ones that are capable of producing disease in humans. But that's not the only type of biohazard. Okay? There's also animal biohazards. These are the ones that make our animals sick. There's also plant biohazards. These are the ones that make our trees sick and our flowers sick. We have them all here at OSU. We have a large plant program where they study plant disease. They work with plant biohazards. We don't worry about them as much from a human standpoint because they don't affect us, but if they get out in the environment, bad news. You could kill all the trees depending on what type of biohazard they are. Obviously, the animal biohazards, they do affect us a little bit depending on what they are because we don't want to take them home. If it's something that could affect our pets, our cats or dogs, or hamsters, our gerbils, we don't want to carry those home with us because we certainly don't want to, make our pets sick. Some of them cross over. Some of them make humans and animals sick. Salmonella is a good example of that. Salmonella is a human pathogen. It's also an animal pathogen. Makes horses very, very sick. Makes humans pretty sick too. If you've ever had salmonellosis, which I have, it is awful. I would not wish that on my worst, well, maybe my worst enemy, but not somebody that I just dislike. Where do we find them? Clinical or research labs where we've got culture stocks, bodily fluids or tissues of human or animal origin, pretty much everywhere here on campus where we're doing research or in the hospital. Biohazards includes things like bacteria, fungi, viruses, parasites, recombinant DNA, prions, even things like animal allergens, animal dander, what else do we have? These are the main ones. So we talk about biohazards and then we talk about biosafety. These are not the same thing, okay? Biosafety is the application of knowledge, techniques, design, and equipment to prevent personal, laboratory, and environmental exposure to potentially infectious agents or other biohazards. So biosafety is kind of the science of working with biohazards. Biosafety is relatively new. When I say relatively new, I'm talking maybe 50 years. And there aren't a lot of programs where you can go and study biosafety. You don't find a lot of people that go get a PhD in biosafety. There just aren't a lot of programs out there. There are a few, but there's not a lot of universities that you can go and get a master's or a PhD in biosafety. Biosafety is kind of a compilation of a lot of different sciences. So it's not like chemistry or microbiology or physics where you just go and study that discipline. It's a lot of things. It's engineering, it's microbiology, it's a little bit of chemistry. All those things put together to learn how to work safely with these agents. Why do we practice biosafety? Well, our program goals here at the university and anywhere else where you're working with these agents are to protect our staff, our students, and our visitors, number one. That's our biggest goal. Second, we want to prevent environmental contamination. Thirdly, we want to protect our experimental material. We want to make sure we don't contaminate it. We want to get good experimental results. And then, of course, we need to comply with federal, state, and local regulations. Acquiring infections from working with infectious agents is a recognized and documented occupational hazard. So if you are an employee and you have an exposure to an infectious agent and you become sick, that is actually an occupational exposure under the OSHA standard. And it gets reported in our OSHA 300 log. If you were very ill or missed work, it qualifies for workman's comp, just like if you fell down a flight of steps or broke your hand, hit it with a hammer when you were a maintenance guy. So this is a documented occupational hazard. This right here, what we're going to talk about next, is extremely important. Obviously, I don't want you to only learn one thing.